All right, guys, in this video series, we're going to be doing an introduction to the immune system. And I like to set this up as thinking about the goals of what your immune system is trying to achieve. So the first option is just never let bacteria or viruses or other pathogens even get inside of you in the first place. So option number one is ideal, right? You've got skin, you've got stomach acid, you've got mucous membranes. All of this is fairly straightforward, and so I'm going to skip over this for the purpose of the video because it's probably things that you've seen in a physiology class before. All right, option two is where things start to get a little more interesting. So if bacteria or viruses get past your physical barriers, right? They enter your skin, get in your respiratory tract. They're actually inside the body now. We want to recognize that pathogen and try to destroy it by recognizing general properties of those pathogens. And when I say general properties, what we mean by that is, at this point, you haven't made antibodies, you haven't activated the adaptive immune system yet, and so it's not quite as easy to do the recognition part of this. So what are we going to do? You're going to activate nonspecific cellular responses, which are all part of the innate immune system, right? And when you see that term innate, all that's really telling you is it's genetic, it's encoded. There's nothing that can really change or evolve over time. Like, you've got what you were born with. And so the first step in that process is inflammation. And inflammation gets a bad rap because it has pain that comes along with it. But it's actually a good thing as far as the healing process goes because, one, it increases blood flow. All right, this is why tissues turn red. You get vasodilation of arteries and arterioles leading into the injured site because we want to get more white blood cells and more nutrients to the site to start the repair process. Right? And in addition to that, white blood cells at the site of the infection are going to release chemical signals. Right? You'll often see these referred to as cytokines. And those cytokines really are just chemical attractants to other cells. And that's what this is showing. This is the outline of a blood vessel, right? A capillary. And this is a white blood cell, probably something like a monocyte or a neutrophil, squeezing its way through the wall of the capillary to invade the surrounding tissue, right? That's how inflammation actually kicks off the healing process. Okay. In addition to that, there are going to be some systemic things that happen too. You're probably going to get a fever if the infection is significant. And the idea here is fever is kind of the same concept as inflammation. It's seen as a bad thing because it's a sign that there's an infection going on. But assuming you have an infection, a fever is an expected response, right? It's supposed to happen. And so if the fever is on purpose, the question is why? Why is that beneficial? Well, bacteria and some viruses replicate best at normal body temperature. And so if we raise that above normal, it makes it more difficult for the pathogen to divide and can slow down the process. All right, it's also a good sign that your immune system is actually paying attention. In this clinical trial, some very interesting data. They took people that had a beta hemolytic strep infection, which is pretty awful. This is an infection in the blood, right, bacteremia. And these people are at severe risk of dying. And you can see that because in their cohort, 272 people survived, but a pretty significant portion, 42 of those, didn't survive. And the question they asked was, do any of these things, confusion, pain, consciousness, fever, or dyspnea as difficulty breathing, did any of those symptoms predict who did better or who did worse when they had the infection? And this odds ratio, this is a concept from statistics where 1 means it's average. And so if the odds ratio comes out as 1, it means it doesn't matter. It doesn't matter if you do or don't have that symptom. It doesn't make a difference. If it's above 1, it means that you're at increased risk. And if it's below 1, it means you're at decreased risk. And so what we can see from this is people that were confused, an odds ratio of 3.3 means that they were 3.3 times more likely to die than someone that didn't have confusion. Right? Same thing, unconsciousness, even worse, right? 6-fold increase in mortality. Difficulty breathing, also an increase. What's cool, though, is 2 symptoms, pain and fever, were actually negatively correlated, right? 0.5 means there's half the chance of dying as normal, and fever means 0.2, so 1/5 the odds. all that's saying is pain and fever might not be very pleasant, but they're a sign that your immune system is doing what it's supposed to. If you have a horrible infection like beta hemolytic strep and you don't have pain or a fever, clearly your immune system is dropping the ball and it's not really paying attention to what's happening. All right. Okay, so now that we have inflammation, we're getting some systemic responses like fever. We need to get nonspecific cellular responses going, right? And the first part of this is we need to kick off the process of phagocytosis. Phage in Latin just means to eat, and cyto is cell. So phagocytosis really just means cellular eating. You have a handful of white blood cell types, neutrophils, monocytes, and when monocytes leave the blood, we call them macrophages. Those aren't the only phagocytic cells in the body, but they're definitely the most abundant. So here's what we need to happen. We need your phagocytic cells to recognize that there's some sort of pathogen present, right? In this illustration, this green sphere is supposed to be the pathogen. You can imagine it being a virus or a bacterium. It doesn't really matter, okay? In order for this cell to recognize the pathogen, it has to detect some sort of molecule on its surface. And since we don't have antibodies made yet, it has to be something reliable, right? And so this cell has to detect some sort of molecule that's specific to bacteria or viruses or other pathogens. And so we call these pathogen-associated molecular patterns, right? And all that means is molecules that are very, very highly specific to pathogens that you would never find on human cells, right? And so it's an indicator that if those phagocytic cells bind any of these molecules on their receptors, that they're going to say, all right, we better phagocytize and destroy whatever's on the other end of that molecule because it's clearly something foreign. So for example, peptidoglycan is only found in the cell walls of bacteria. Perfect pathogen-associated molecule because it's only found in bacteria and it's essential. Bacteria can't very easily just get rid of their peptidoglycan to sneak by your immune system. And so we want these molecules to be important, critical molecules for the pathogen so they can't just evolve the loss of that molecule as a way to hide from the immune system. So peptidoglycan, LPS in bacteria, mannose is a sugar that's not found in humans, that is found in bacteria, and double-stranded RNA is used for viruses. Viruses are a lot simpler, so there aren't as many of these pathogen-associated molecules to go around for them. Okay, but the main point of this is if your phagocytic cells recognize any of those pathogen-associated molecules, right, that's what these receptors are binding to, and that is what triggers phagocytosis. Okay, once that happens, that just means that the pathogen is being ingested inside of the phagocytic cell, and you get the development of the specialized organelle called a phagosome, which is a lot like a lysosome. It's full of digestive enzymes similar to what you would find in a person's stomach and reactive oxygen species that destroy whatever's on the inside. And so your pathogen is really just going to get chopped up into pieces. And since bacteria and viruses are made out of the same components that human cells are, like proteins and nucleic acids and sugars, it's easy. We just break those down into their component molecules, and they can get recycled and used throughout the body. All right. In addition to that, there are a couple more non-specific responses to tag on here that go along as kind of an adjunct to the rest of what we've been talking about so far with phagocytosis. One of those things is a signaling process called the complement system. And complement got its name because it's complementary to the use of antibodies. And there are a few main effects. One is those complement proteins, so all of these things, C2b and C3 and C5, those are just different complement proteins. There are a bunch of them. And they're made by the liver, they're secreted into your blood, so they're there all the time. And they serve as secondary receptors to pathogen-associated molecules to make it easier for your immune system to detect them. And they can bind to antibodies that are attached to antigens, right, on your pathogen. And so complement because they complement and amplify the effect of antibodies. So the end result is if complement proteins stick to the pathogen, one, it attracts phagocytic cells. Two, it amplifies the effect of antibody binding. And three, it can also form a protein complex, right? This it's sometimes called an attack complex, and it very literally pokes holes, right? Forms pores in the membranes of bacterial cells or other pathogens, and makes their cytoplasm leak out, and so it can kill bacterial cells directly as well. All right. Okay, so complement is most effective for bacteria just because they have more pathogen-specific molecules, and they have cytoplasm that can actually be damaged by that attack complex. All right. Okay, last one, interferons. This is more specific to viruses, and interferon is basically a chemical signal that's released from a virus-infected cell to let other human cells that are nearby know that there's an infection going on. And so in the example in this figure, this cell, they're illustrating it being infected by COVID, but it could be any virus theoretically. Once that viral infection starts happening, because remember, viruses are only intracellular parasites. They have to get inside of a human cell. Once that happens, this cell will start activating this interferon pathway, which really just means that it makes these little protein signals called interferon molecules that are released from the cell, right? These are your interferons, and they bind to interferon receptors on neighboring cells. Okay, that tells this neighboring cell that the warning system has been activated, there's a viral infection, and the end result of this pathway is those cells do things to try to reduce the impact of viral infections. So they make enzymes that chop up double-stranded RNA, which is only found in viruses, not humans. And they can start blocking translation, meaning turning off ribosomes. And that's not something you would want to do long-term because the cell would die. But for a day or two, to ramp down translation makes it harder for the virus to make copies of itself, harder for the virus to spread. Okay. All right, so in summary, all the mechanisms that we just went over, these are parts of the innate immune system, right? They're nonspecific. We haven't made antibodies yet. We haven't started activating B cells and T cells, but that's going to come down the road in a little bit. So in the playlist, follow through to the next video, which is genetic resistance to pathogens, right? Avoiding infection for genetic reasons, not really an immune response. Okay, so flag the playlist, watch them in order. It'll make a lot more sense that way, and subscribe to the channel so you get all the updates when new videos are released.